Imagine a world, a not-so-distant future perhaps, where the very definition of family and parenthood is rewritten. A world where the biological constraints that have defined reproduction for millennia are challenged and overcome. Imagine a world where a baby could have two biological dads, a concept that once belonged solely to the realm of science fiction, both contributing their DNA, their unique genetic blueprint, to create new life. A life that carries the combined heritage of two fathers, blurring the traditional lines of inheritance and lineage. For ages, biology insisted this was impossible. The fundamental laws of nature, as we understood them, dictated a different story. Mammals, by definition, need one egg, the female gamete carrying half the genetic material necessary for life, and one sperm, the male gamete, contributing the other half to complete the genetic puzzle. This union, this dance of life, was considered immutable. But in 2018, a groundbreaking moment arrived. Scientists in China, through years of dedicated research and relentless experimentation, shattered that rule. They dared to challenge the seemingly unchallengeable by creating the first mouse born from two male parents. A tiny creature, yet a giant leap for science. This wasn't a fluke, a lucky accident in the lab. It was the result of years of painstaking genetic research, a deliberate and methodical pursuit of a seemingly impossible goal, and precise planning, meticulous execution, and an unwavering commitment to pushing the boundaries of what we thought was possible. The mouse, healthy and normal, defying all expectations, was living proof that the building blocks of life, the very essence of our being, could be assembled in a revolutionary way, challenging our fundamental understanding of genetics and reproduction. Its birth sparked global conversations, igniting debates and discussions within the scientific community and beyond, about the future of families, the evolving definitions of parenthood, and the potential for new family structures to emerge, and the ethics of genetic technology, forcing us to confront the moral implications of manipulating the very code of life. Could this breakthrough one day help same-sex couples, particularly male couples, have children who share both their genes? A child that is truly a blend of both their heritages, a dream for many. The story of this little mouse, a seemingly insignificant creature, is more than a scientific milestone. It's a testament to human ingenuity and the relentless pursuit of knowledge. It's the start of a new chapter in how we think about life itself, a chapter filled with possibilities and profound ethical considerations. Let's dive into how this was possible, exploring the scientific breakthroughs and the intricate techniques that led to this extraordinary achievement. So how did scientists manage to pull off the seemingly impossible? Creating a healthy mouse from two dads. It sounds like something straight out of science fiction, doesn't it? But this groundbreaking achievement is very real, and it opens up a whole new world of possibilities in the field of genetics and reproductive biology. It was like following a complex recipe, a meticulously crafted protocol that demanded precision and a deep understanding of the intricate workings of DNA. Think of it as baking a cake, but instead of flour and sugar, you're dealing with genes and chromosomes. And this recipe called for some wild ingredients, unconventional components that pushed the boundaries of what we thought was possible in mammalian reproduction. These weren't your everyday lab materials. They were specially engineered cells and carefully selected genetic components. They started with sperm from one male, the conventional starting point for creating offspring. But this was just the beginning of a far more complex process. And then they introduced a special stem cell from the other male, a unique type of cell with the potential to develop into various tissues. This stem cell held the key to overcoming the biological barriers that typically prevent same-sex reproduction. This wasn't just any stem cell, it was one that carried only half the usual DNA, a haploid cell. This characteristic was crucial for the next steps in the process, allowing the scientists to manipulate the genetic material in a specific way. Next, they took an egg from a female mouse, a seemingly paradoxical step in creating offspring from two males. But this egg served a very specific purpose in the overall procedure. They carefully removed its DNA, essentially erasing the female genetic contribution from the equation. This step was essential to ensure that the resulting offspring would inherit genetic material solely from the two male parents. The egg, now devoid of its own genetic material, was used as an empty vessel, a blank canvas ready to receive the genetic blueprint from the two fathers. This enucleated egg provided the perfect environment for the subsequent genetic manipulation. Into this vessel, they precisely placed the DNA from both fathers, combining the genetic contributions of the two male parents into a single cell. This delicate procedure required advanced micro-injection techniques and a steady hand, but there was a catch, a significant hurdle that had to be overcome to ensure the successful development of the embryo. This wasn't just a simple matter of combining DNA, there were other factors at play. You see, our genes have imprinting tags, epigenetic markers that play a crucial role in regulating gene expression. These tags act like labels, indicating the origin of each gene. These imprinting tags tell them if they're from mom or dad, influencing how the genes are expressed and ultimately affecting the development of the offspring. This parental origin information is vital for normal development. So to make the embryo develop properly, the scientists had to address this imprinting issue. Without the correct imprinting, the embryo would not be able to develop normally. The scientists had to edit the stem cells' tags, manipulating the epigenetic markers to ensure the correct gene expression patterns. This required precise and targeted gene editing techniques. They had to modify the imprinting tags to mimic a mother's DNA, essentially tricking the embryo into thinking it had inherited genes from a female parent. This was a crucial step in overcoming the biological barriers to same-sex reproduction. This gene editing tricked the embryo, fooling its developmental mechanisms into believing it was a product of conventional reproduction. And this manipulation allowed it into growing normally, bypassing the developmental roadblocks that would have otherwise prevented its survival. The embryo began to develop as if it had a mother and a father. The successfully manipulated embryo was then implanted, carefully placed into the uterus of a surrogate mother. The embryo was implanted into a surrogate mother, a female mouse who would carry the developing offspring to term. The surrogate mother then carried it to term, providing the necessary environment and nourishment for the embryo to develop into a healthy pup. Of course, this wasn't a foolproof process. Out of many tries, after numerous attempts and refinements to the technique, the scientists finally achieved success. And finally, a few healthy pups were born, a testament to the ingenuity and perseverance of the research team. These pups represented a major breakthrough in reproductive biology. What's even more remarkable is that these pups even went on to have babies of their own, demonstrating that the procedure didn't compromise their reproductive capabilities. This confirmed the long-term viability of the technique. This groundbreaking achievement proved the process could work, opening up new avenues for research and potentially revolutionizing our understanding of mammalian reproduction. However, it's important to remember that this was far from easy. The process involved intricate manipulations and a deep understanding of complex biological mechanisms. So this begs the question, what exactly is gene editing? This powerful tool that allowed scientists to rewrite the genetic code. And how did it make this seemingly impossible feat possible? How did scientists use this technology to overcome the natural barriers to same-sex reproduction? Gene editing is a revolutionary technology, a bit like using a high-tech find-and-replace tool. But instead of words, we're working with the very building blocks of life. It allows scientists to precisely alter the genetic material of living organisms, opening up possibilities we once only dreamed of. This find and replace operates on the DNA instruction manual found inside every living cell. Think of it as going into the source code of life itself. DNA is the complex code that meticulously tells our bodies how to grow, develop, and function, dictating everything from our eye color to our susceptibility to certain diseases. It's the blueprint of life. Within this code, genes are the specific words or instructions that determine particular traits or characteristics. They are the functional units of heredity. With gene editing, scientists gain the unprecedented ability to precisely cut into this DNA sequence, fix errors or mutations that cause disease, or even swap out specific genes with modified or corrected versions. Just like editing a document, but on a microscopic molecular scale. It's like debugging the software of life. This groundbreaking technology holds immense potential, allowing us to correct genetic diseases at their source, offering hope for cures and treatments that were once unimaginable. Beyond medicine, it can improve crops, making them more resistant to pests and diseases, and increasing their nutritional value. And, as demonstrated by the two-father mouse, it can rewrite the fundamental rules of reproduction, challenging our understanding of what's biologically possible. In the remarkable case of the two-father mouse, scientists had to meticulously change the imprinting tags, which are epigenetic markers that determine how genes are expressed. These tags were altered on one father's DNA, to essentially trick the system into thinking it came from a mother, a feat of genetic manipulation. Without this incredibly precise editing, the embryo simply wouldn't have been able to develop and grow. The imprinting tags are crucial for proper development. Gene editing made the seemingly impossible possible. It overcame a fundamental biological barrier. This allowed life to begin in a way that nature, in its ordinary course, never intended, pushing the boundaries of scientific understanding and capability. It's an incredibly powerful tool, offering unprecedented control over the genetic code, but it comes with big responsibilities, demanding careful consideration of the ethical implications and potential consequences. The revolutionary tool that made this particular breakthrough possible, and is at the forefront of gene editing technology, is called CRISPR. So let's take a closer look and see how CRISPR actually works at the molecular level. CRISPR is the revolutionary molecular scissors that made gene editing fast, cheap, and precise. It works by pairing an enzyme called Cas9 with a guide RNA that leads it to the exact spot in the DNA to cut. Once CRISPR finds its target, Cas9 snips the DNA and the cell's repair system takes over. Scientists can use this moment to disable a gene, fix a mutation, or insert new DNA. In the two-father mouse experiment, CRISPR was used to delete 7 specific imprinting tags, making male DNA behave like female DNA. Before CRISPR, this kind of genetic surgery was nearly impossible. Now, scientists can rewrite the rules of reproduction with unprecedented accuracy. This technology is transforming biology and medicine worldwide. But with great power comes the need for caution. So what does this mean for families and for the future? For many, the two-father mouse is more than a headline. It's a symbol of hope. Same-sex male couples have long dreamed of having a child genetically related to both partners. This research hints that, one day, it might be possible. The technique could also help people facing infertility, offering new ways to create healthy embryos. But the science is still in its infancy, and there are huge hurdles ahead. For now, it's a proof of concept, a glimpse of what might be possible in the future. But why is this process so difficult, and what challenges remain? Let's take a closer look. Creating a two-father mouse was a massive challenge, with many failures along the way. Most embryos didn't survive, and many pups had health problems. Editing the right imprinting genes is like performing surgery on a microscopic scale. One mistake, and development fails. The special stem cells used are tricky to make and keep healthy. Even CRISPR, while precise, can sometimes make accidental cuts, leading to unpredictable results. The health and safety of any future child must come first, and right now, the risks are too high for humans. Scientists have a lot more to learn before this could ever be considered safe. And beyond the science, there are even bigger questions to answer. Let's talk about the ethics. Every leap in science brings tough ethical questions, especially when it comes to creating life. Is it right to try this in humans when the risks are so high? The well-being of any child must be the top priority. This technology also challenges our ideas about parenthood and family. We need open, respectful conversations about where to draw the line between treating disease and enhancing traits. The debate over gene editing's limits is just beginning. And even if the science advances, the road to human use is long. Turning a mouse experiment into a safe human procedure will take decades. The genes involved in imprinting are different in humans, and every step must be tested in other animals first. Strict laws and regulations currently ban this kind of gene editing in many countries. Even if the science is perfected, society must decide if and how to use it. The journey ahead requires caution, transparency, and public trust. The scientists behind this work stress that their goal is understanding biology, not rushing to human trials. So what does the future hold? The two-father mouse is a symbol of possibility and progress. It shows that the rules of biology can sometimes be rewritten, opening new doors for science and families. This research could one day help same-sex couples, people with infertility, and even lead to new treatments for disease. But it also reminds us to move forward with care, wisdom, and open dialogue. The future of family and of science will be shaped by our choices today. With curiosity, responsibility, and hope, we can use these breakthroughs